Hello! Today we're going to be talking about our first kingdom, Kingdom MOnera. So this is showing you a blood agar plate which you can plate bacteria on, and we're going to actually grow our own bacteria. I want to point out that this is considered a lawn of bacteria. It's very concentrated. So when you first start planting a bacteria, you take a cotton swab and wherever you think bacteria is going to be growing, and then you're going to rub it in this direction, and then you pull down in this direction, pull down in this direction, and this direction. And as you do all the dragging, you kind of dilute the bacteria so you can see the individual bacteria. So bacterium is a singular form, where bacteria is plural. It's derived from the Greek word meaning monorans, which is single. It refers to a single cell nature. And there are three main shapes that we'll talk about. You have circular, rod, and spiral. Okay. This is what it would look like. Now, that's crazy. You don't have to write this down, but I just feel this is so important. There's more bacteria that inhabit a handful of dirt or your human mouth than the total number of people who have ever lived. So they are impressive. Their collective biomass is at least 10 times that of eukaryotes. Okay, so they are all prokaryotes, okay? They appeared about 3.5 billion years ago, and they lived and evolved all alone on Earth for about 2 billion years. Again, prokaryotes means no nucleus or membrane-bound organelles. They are the simplest of all life forms, okay? They are found everywhere. Air, soil, water, surface of your desk, your skin, inside your body. There's about 10,000 known species. Prokaryotes outnumber all of the eukaryotes combined. Okay. And just again showing you the difference. When we talked about the cell, we showed you this picture. So you have a bacterial or prokaryotic cell and then the eukaryotic cell. Okay. So we can break down bacteria into two different groups. All right. We have what's called the gram staining. Okay, this is a very valuable tool in identifying bacteria. It's used to separate bacteria into two groups based on their difference in their cell wall. And we have two types. We have gram positive and we have gram negative. So gram positive have simpler cell walls. They have relatively large amounts of peptidoglycan, and it's going to stain purple. So P positive purple peptidoglycan. Okay, and they can be pathogenic. Not all the time, but they are typically pathogenic. Okay, so gram-positive bacteria stain purple, have a lot of peptidoglycan, and they can be pathogenic. Gram-negative has a more complex cell wall. It's going to protect the bacteria from some antibiotics. They're going to have less peptidoglycan, and they're going to have lipopolysaccharides, which are carbohydrates that are bonded to lipids. There are also many disease-causing bacteria. They tend to be more threatening than the gram-positive, okay, because the lipopolysaccharides are often more toxic. And they are commonly more resistant to antibiotics because of their cell membrane impedes the entry of drugs. And it will stain red. So think of negative as bad and it will stain red, and again, it is a little bit more harmful. Okay. Okay, this is just showing you the cell wall and staining red. So gram-negative stains red, has less peptidoglycan and lipopolysaccharides, which typically is a little bit more threatening. Okay, so size is small, about 1 to 5 micrometers, and I'm just giving you an idea a little bit about the size. So again, you don't need to write that down. They're larger than the viruses, but they're still pretty small, and we do need a microscope to see. Okay, so their structure, they're prokaryotes, so they do not have a nucleus nor membrane-bound organelles. They do have ribosomes. The ribosomes are slightly smaller than the eukaryotic version, and it does kind of differ in the protein and the RNA content. Okay, the cell walls are made up of peptidoglycan and also the lipopolysaccharides, and that helps to protect and maintain that osmotic balance, helps to maintain the shape of the cell. Penicillin, however, can destroy those cell walls. and that was going to inhibit the synthesis of cross-links in the peptidoglycan, and that prevents the formation of the functional wall. Okay, so the peptidoglycan is a polymer of modified sugars cross-linked by short polypeptides. So very different than the plant cell wall. Okay, the cell wall can also be surrounded by a capsule, which is additional protection. It's a sticky substance that allows them to adhere to their substrate, allows them to live in colonies together. It allows for nutrient storage, protection from drying out, or escape from the protective defenses that would eliminate bacteria from the host. So without capsules, bacteria that cause pneumonia would not make us sick. All right, so our genetic material in the prokaryotes. It does not have as much genetic material as a eukaryote. It has about 1/1000th of much DNA as a eukaryotic cell. And again, it's one strand. It's concentrated in a nucleoid region that will stain a little less dense than those of the cytoplasm. So it's not like that. And again, you have one double-stranded DNA molecule. You can have plasmids, which the plasmids are those circular pieces of DNA that does contain a few genes. Bacteria can survive without their plasmids because all of the essential functions are actually found in that one chromosome. But the plasmids have genes that allow for resistance to antibiotics, metabolism of unusual nutrients not present in the normal environment. And you can replicate that independently of the main chromosome. And that is why they're used in the genetic research. Plasmids can be transferred between partners when the bacteria reproduce or conjugate. Okay, so again, plasmids only consist of a few genes. You could survive without them. They replicate independently of the chromosome, and they can be transferred between partners during conjugation. This is just showing you a little bit about the structure. Okay, you have the nucleus and the chromosome in the nucleoid region. They do not have a nucleus. You do have cytoplasm. You do have free ribosomes. You have a cell membrane, a cell wall, a capsule, and then we have some structures that help it move. There are three main types of bacteria. You have round or coccus, rod, bacillus, and spirillum or spiral. Okay, so coccus is round, bacillus is rod, spirillum is spiral, and there's a couple more examples. They're vibros, it looks like commas, spirochetes are longer, more flexible. There actually, in 1980, there was a square bacteria that was found in a salt pond along the Red Sea. It was called the genus Arcula. And then there was a flower petal-shaped Simoncelia, which were combined to form a cluster resembling a rose. Okay, but we're just going to focus in on the three main types. So this is coccus, bacillus, spirillum. Okay, so diplo means two, strepto means chain, staphylo means cluster. So diplococcus is two round bacteria. Streptobacillus is a chain of rod-shaped bacteria. Staphylococcus is a cluster or a bunch of aggregated together of round bacteria. All right, so endospores are pretty impressive. Okay, this is a highly resistant dormant structure that is formed when conditions are harsh and unfavorable. So there's a protective wall that will surround the DNA and little cytoplasm. Okay, so the original cell will replicate its chromosome. One copy becomes surrounded by that wall, and that tough protective wall around the DNA and the cytoplasm will withstand long periods, even centuries of boiling, years of freezing and drought, lack of nutrients, water, UV light, poisons. Okay, it is quite impressive. They can hydrate and revive in hospital conditions, though. They are resistant to the effects of aging. Excavation of dormant spores remained alive after 1,300 years of suspended animation. So it's a very cool adaptation that bacteria have. And again, it's just a protective wall around DNA and a little cytoplasm. Okay, so bacteria can move or not move. All right, so if they move, they have flagella, cilia, pili, Spirochetes move, or they could secrete slimy chemicals, or they don't move. Okay. The flagella is made up of microtubules, a little different than the flagella of the eukaryote. It's about 1/10 of the width of eukaryotes, and they are not covered by the extension of the plasma membrane. So they're very unique in structure and function. Okay, the spirochetes have several filaments spiraling around the cell underneath the outer sheath of the cell wall. It's very similar to the flagella in structure, but it kind of allows for a movement like a corkscrew. Cilia, again, are those little tiny hairs that are very numerous. Pili are a little more intermediate in size, and they kind of allow the bacteria to inch. Okay, the secreting slimy chemicals allow the bacteria to move by a gliding motion, and it kind of helps it to move if it doesn't have the flagella and so forth. Okay, so the next up, that's just showing you the cilia moving. The pili is a little more intermediate. Okay, reproduction of monerans. Okay, they can reproduce asexually through binary fission, which is simply dividing the two cells of equal size. This may take a few minutes. Okay, they also can form the spores or the endospores, and again, they can withstand many years and they don't age. Or they can go through sexual reproduction, which is conjugation. Conjugation is the exchange of genetic material. A long bridge of protein forms between and connects the two bacteria. Remember, bacteria can't form mitosis because there's no nucleus. So no mitosis nor meiosis occurs. But bacteria can double every 20 minutes, like E. coli. It could take 1 to 3 hours. Okay? So it's quite rapid growth as long as there are enough materials and resources available. Again, conjugation is sexual, exchanging genetic material. Binary fission is just the splitting of the cell. Okay, so bacteria need certain things in order to grow. Okay, temperatures. They typically like the temperatures between 80 and 100 degrees. Okay, refrigeration will retard the bacteria growth, so that's why we put food in our refrigerator so we don't get bacteria. They need some type of moisture. Light, dark encourages growth, where UV light kills. They need nutrients. Their pH needs to be a certain way, and they need salt concentrations that are certain, specific to their species. So all conditions will vary within the species. Okay, so when we first started out with life, the first bacteria, the bacteria did not have any oxygen around. So there are 3 conditions for oxygen growth. Obligate aerobes use oxygen for cell respiration and cannot grow without it. It's an obligation to be aerobic. Facultative anaerobes can use it if it's present, but it's okay if it's not. Obligate anaerobes means it's an obligation to not have oxygen. It is poisoned by oxygen and will die. Okay, so they could be autotrophs or heterotrophs. If it's an autotroph, it could be photoautotrophs or chemoautotrophs, and heterotrophs could be photoheterotrophs or chemoheterotrophs. Okay, so for the phototrophs, okay, they use light energy. Okay, photoautotrophs harness light energy to drive the synthesis of organic compounds from CO2, like the cyanobacteria. Photoheterotrophs use light to generate ATP, but must obtain their carbon in organic form. Chemotrophs obtain their energy from chemicals taken in from the environment. So a chemoautotroph is chemosynthetic bacteria. They need CO2 as their carbon source. Okay? Chemoheterotrophs must consume organic molecules for both energy and carbon. And the majority of bacteria are this chemoheterotrophs. Okay, we're going to have you go to this next PowerPoint just to finish up. There's not much more to watch, but again, I have a limit on 15 minutes. So please watch part 2 of this, and then we will continue on. Thanks.